Awesome. Thank you so much, everybody. Welcome. My name is Jeremiah Dixon. I am a third-year neuroscience major studying migratory cell tracing, in which I will be investigating magnetotactic bacteria, MMS6, for MR reporter gene viability. I have the privilege to be working under the tutelage of Dr. Xiaoping Hu, my respective faculty mentor, who works in the bioengineering department, as well as Abby Barlow, and Tong Lu Du. So without further ado, let's go ahead and jump into the heart of this project. Let's see here. Understanding the fundamentals of DNA transcription is crucial to gene therapy. Recently, the prospect of magnetic reporter genes originating from aquatic magnetotactic bacteria have been allowing researchers to better image cell migration. Briefly, what MR reporters are, they allow cells to accumulate extracellular iron that intensifies cellular images. This study in particular helps and aims to utilize literature analysis to expound on the current knowledge of MMS6 to provide a preliminary foundation for future MR integration. And before that, I want to go ahead, before the heart of the project, I want to go ahead and talk a little bit about the impact, more significantly, the background of why this project is so significant. Once optical reporters, that is green fluorescent protein tags, were the hallmark for cell tracing, and they're extremely crucial for deducting gene function. Notably, the main reason why gene function is so necessary for bioengineering is because in gene function, we are allowed to analyze triggers that set off disorders such as cancer or neurodegenerative diseases. However, because of the intrusivity of the optical reporters, in which they would have to do small surgeries on animals or plants to inject these GFPs, researchers looked for a more viable and a more efficient way for imaging cell groups. They found this answer, thankfully, in MR reporters, which are magnetic reporter genes, genes which respond to the applied magnetic field of an MRI to give researchers an idea of the biochemical reactions within that cellular environment, and as well the cell tracing of that particular specimen. So we've divided our project, my project, into three phases. Phase one, optimized concentration. In this phase, we are currently culturing the cells and measuring them in the MRI at the Center for Advanced Neuroimaging here at UCR. In this, we hope to do a controlled R2 analysis, that is the relaxation state of in which we are able to analyze the iron concentration, in which that enables us to quantify how successful the MMS6 is in responding to that magnetic field, and giving researchers an idea of how strong and how viable this is if we were to put it into cells to track, say, cancer or hypoxic conditions. So in figure 1A, if I can turn my laser on, figure 1A, this shows a brief overview of what a magnetotactic bacteria is. Essentially, there are iron fragments that float outside the cell, and these iron fragments are encapsulated by the inner membrane of the MMS6, and a lot of these encapsulations lead to a creation of what we call a magnetite crystal. These magnetite crystals are aligned along a magnetosome chain backbone, which, as we see here in figure 1B, give these bacteria the way to navigate the Earth's waters for better growth conditions, and something that is actually very interesting to study for biologists in particular. But anyway, this whole mechanism of magnetic interactions in bacteria have given way for us to be able to trace cellular interactions. Thus, here we are with MRI imaging and controlling it analysis, the R2 analysis of the signals and the viability for biomedical engineering. The next phase, phase two, in which we'll be talking about, is the tumor modeling. Now, phase one talks a little bit more about how we optimize the concentration, how we get an ideal growth for these MS6 genes. The next phase, we apply a real life problem, the problem of tumors. Tumors have been extremely deadly to researchers in the fact that the more you are able to, the earlier you're able to detect them, the better you are at preventing them. However, researchers have not been able to detect them as early as they hope, thereby drastically affecting survival rates for things such as cancer, and so on and so forth. In this phase two, we hope to do an experimental R2 analysis measuring the signals of the MS6 under a tumor inoculation. Below here, figure 2A is the image by courtesy of Emory University, in which they inoculated two tumors, 9L respectively and 9L4S, into a mouse's hindsight. And in that, they injected MMS6 right here, and were able to get a reading that was able to allow researchers a little bit of an idea to how that tumor was reacting with its cellular environment, and also trace its migration. These two images right here show the cell growth, one down here and one down here. As you can see, the cellular interaction is a lot more pronounced. This giving the researchers the idea that the cell of the tumor is actually growing, and the MMS6 is able to track it along its progression stage. So this is the hallmark of what we want to do here at UCR. We want to replicate this entire experiment for ourselves for reproducibility, so that we can be able to incorporate that into further research, in which leads me to phase three, the hypoxic model. The hypoxic model. First, we have phase one, optimized concentration. Phase two, the tumor model, and now phase three, another real life problem that bioengineers face, in which we'll be doing an experimental R2 analysis of iron concentrations in low oxygenated environments, or low oxygenated hypoxic. These resemble the neurodegenerative disorders such as Alzheimer's, that plague a lot of the American population, and especially ischemic strokes, and a lot of other things. Researchers hope to trace and analyze. Again, early detection is better for prevention, and the earlier that we can detect these things, the better we are at preventing them. Hence, we are applying MS6 into a resemble real life problem. In this, we'll be doing essentially a low oxygenated environment and injecting cells that have been inoculated with MMS6 into those regions, and obtaining images like these for future reference that can be enabled to expound the former and current knowledge of MMS6 as an MR reporter gene. In summary, the MTB, magnetotactic bacteria gene MMS6, is from a ferritin-based or iron-based MR reporter. The significant impact that this has to be able to produce autonomous iron on itself has been desired by researchers to be desired by researchers all around for intracellular investigation of migratory cell tracing. So with that, I'll go ahead and end. Thank you so much for listening. If you have any questions, please ask. I'll open up the floor right now.